Welcome to the CanMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amaralt. I am the Senior Marketing Manager at Medicinal Genomics and proud member of the team that puts on the CanMed Summit. Okay, I don't have too many updates for you today. We're still working on getting the CanMed 25 videos and photos edited and posted, but those should be live before our next episode. We will email everyone on our list when those are live, so if you're not on our list, please go to canmedevents.com right now and sign up for email alerts. We are also still finalizing the details for our CanMed 26 Summit location and dates, which we also hope to announce very soon. Our guest today is Dr. Jose Leme. Jose is a professor at Southern Illinois University, and he's the principal investigator in the Cannabis Biology and Cultivation Systems Lab. He and his team focus on determining how to leverage environmental controls such as light intensity, duration, quality, temperature, and CO2, as well as plant nutrition, biostimulants, and hormones to improve plant production. Moreover, the research group is developing techniques to optimize plant yields and quality based on key crop steering traits that increase sustainability and efficiency via plant physiological, biochemical, and agricultural approaches. During our conversation, we discussed several recent studies that Jose and his team have published, including using mechanical stimulation and high-stress training to increase production of secondary metabolites, applying plant hormones to increase trichome density, assessing how drought stress affects plant yield and potency, standardizing cloning methods for plant production, developing standards for measuring trichome density, and more. Before we get to my conversation with Jose, I'd like to thank this episode's sponsor, Advanced Nutrients. Founded in 1999, Advanced Nutrients was the first to develop a complete nutrient system that unlocks the true genetic potential of the cannabis plant. Since its inception, the brand has introduced more than 50 innovations in the cultivation community and continues to revolutionize the space through proprietary scientific discoveries. Learn more at advancednutrients.com. Okay, and without any further ado, please enjoy my conversation with Dr. Jose Leme. Good afternoon, Jose. Thanks so much for joining us on the podcast.
Thank you. We're happy to be here.
Yeah, I am excited to talk with you today about, well, a lot of different things. You and your team at Southern Illinois University have been busy publishing all sorts of information related to improving cannabis plant production and standardizing protocols. So I'm excited to talk with you about that. And some of that information you shared with us at CanMed 25. We're happy to have you there. You were on a panel about pest management and remediation. You also presented a poster about thigmomorphogenesis. Did I say that right?
Yeah, I had to practice this word a lot. So, but yeah, that's correct.
All right. It's a topic that's as interesting as that word is hard to say. So I'm happy to get into that with you. But before we get started, I was hoping you could just tell me a bit about your lab. What's the main focus of your research group and how to get started?
Yeah, thank you for the introduction. I'm happy to be here. I'm sorry for the delay to answer your invitation, but the past 2 months, I'm all over the place. I mean, the past 2 months, I've been in Abu Dhabi, Senegal, Brazil, and Puerto Rico. Then I got back on campus and I was trying to put my life back on track. So but I'm happy to be here. So unfortunately, it took some time for me to answer. But again, I really enjoy CanMed. Was the first time attending it, and I made a lot of connections there. It was an awesome event. So unfortunately, you saw the poster was pretty busy.
Oh
We
yeah.
couldn't even talk, but now it's great to have the opportunity to actually talk to you and be part of this podcast. Um, so talking a little bit about our lab, I'm the principal investigator of the cannabis biology and cultivation systems lab. My position here at Southern Illinois University is divided in two different schools. It's 51% in horticulture and 49% in plant biology. Um, so uh, so it's in the horticulture side, the main focus is control environment agriculture, and in the plant biology side, it's cannabis biology and plant physiology. So it is a good marriage because most of the cannabis production for cannabinoids are indoors, so it's a good
Mhm.
marriage, is a good merge, and so in basically in the control environment agriculture side, we were working with all the CEA, stuff, all the light intensity, duration, quality, CO2, uh, phenomics, plant nutrition, plant biostimulants, hormones. So we are pretty much developing techniques to optimize, cannabis production and quality using, crop steering management or crop steering approach, and then in the plant biology side, we are more focusing in plant physiology. So I do have students, graduate students from in both sides.
Mhm.
So I think we are able to pretty much cover plant production with focus in plant physiology in a pretty good way.
Yeah, and I have to imagine one sort of informs the other, right? As you learn more about the plant physiology, you can refine your techniques.
Exactly, because also control environment agriculture, it can be used as um, um, a production approach, for like grow plants.
Hmm.
But control environment agriculture can also be used for like a research methodology,
Hmm.
because when you're comparing the studies that we're conducting outdoors, the field studies, when you conduct indoors with all this control in environment, the data is much more like clean, and the
Hmm.
results are much more they're much easier to understand. So that's why you can use this approach for like as a production approach, but also as a research approach.
Yeah, no, and that's interesting you say that, and it sort of ties into some of the research that you shared, some of the papers you shared with me that we're going to get into,
Hmm.
sort of standardizing different ways of measuring, or I would assume even sort of the environmental factors that you're looking at too. Because, like you said, if you're comparing one study to another, you need to account for what the environment is like, right? Because that's going to be a huge a variable.
Exactly. And but then when you're using control environment agriculture, you're pretty much standardizing the growing parameters,
Right.
or at least most of the growing parameters like light intensity, spectrum, moisture, temperature. So it's a better way to standardize the research protocols. But again, of course I'm biased because I'm working with control environment agriculture, but
Hmm.
I also enjoy field trials, outdoor studies. But again, I mean, I'm not gonna lie, I'm biased because I enjoy CEA. So
Excellent. And now, is cannabis the main crop that you guys are studying? Is it your sole focus?
um well, I mean, my lab is the cannabis biology and cultivation systems. In theory, the focus is cannabis. But in the beginning, I mean, my program here is pretty unique. In the beginning, I was having a hard time to support the lab because there's not much federal funds
Right.
for cannabis research. I mean, high cannabinoid uh, cannabis research, and uh, and then I start kind of branching out to other plants too, using controlled environment agriculture as a main approach, and then also testing other plants. But now I don't like to talk about politics, but now the funds are pretty bad for everybody.
Mhm.
I do have colleagues freaking out about all this news and this federal funds that are being frozen, and they're like freaking out, and I'm talking to them, and they're like, hey, welcome to my life. It's like I start my lab like exactly in the position you are right now, like no federal funds. So but I was able to support my lab, collaborating with the private sector.
Mhm.
But also, again, it's the main focus, but we are also working with other plants, because of sometimes I have students, very good students, that want to join my lab, and they have a really good knowledge and know-how and experience in controlled environment agriculture, but they're like, oh, my parents don't want me to work with cannabis. Can I test another plant or use another plant? And we are open for that, and you're open for that. So but the main focus is cannabis.
Right. No, and would hemp be any easier to get funding for? Or I mean, at least before kind of politics kind of became what it was or what it is.
Well, I would say for outdoor production and like fiber and grain hemp, it is easier. It is not as complicated, um, but everything kind of naturally pushed me more for the high cannabinoid, even CBD, CBG. No, I'm not even talking about THC,
Hmm.
but the more indoor cultivation of high cannabinoid cultivars. So, because also I guess it's also. Can I talk a little bit about our academic program too?
Yeah,
Yeah.
absolutely.
So yeah, because also my position is divided in research and teaching.
Hmm.
So the teaching part, we are able. Last year, we actually it was approved. It took it's when it when it's addressing cannabis or it's related to cannabis. Anything in academia it takes a lot of time.
Hmm. Yeah.
Um, but last year we approved a cannabis production system certificate, cannabis production system minor, and I think is the first 4 years degree of the entire nation. It's a bachelor degree in horticulture with specialization in cannabis science.
Wow.
So, and then and I'm mentioning that because the horticulture program is really growing because of these new cannabis programs, and I would say 95% of the students, they're interested in the high cannabinoid
Yeah.
uh, approach or high cannabinoid production and management, not really fiber and grain. We do have some faculty members working in the fiber and grain, the cannabis here at SIU. I'm also involved, but most of the interest is for high cannabinoid cannabis.
Excellent. All right. Well, let's get into some of the research that you guys have been doing. And let's start with that thigmomorphogenesis in cannabis. What does that term mean and how can it be applied to cannabis plants?
Cool. Yeah. Thigmomorphogenesis, it's actually a term that was first uh, described in the literature. I think was around 1973, and it's pretty much the um, the effects of mechanical stimulation on plants. And mechanical stimulation is a very broad term because it can be like directly mechanical stimulation. The mechanical stimulation can come from like sounds or other types of vibration. So, but in general terms, um, the description is the effects of mechanical stimulation on plants.
Okay. So what are the effects of mechanical stimulation on
No,
cannabis
yeah.
plants?
So, and again, I mean, and we talk about, I think it's that's another thing that is pretty cool about researching cannabis, because um, again, I mentioned the controlled environment agriculture as a production approach and a research approach, but cannabis, regardless the taboo, it's a really cool plant to be used as a model plant, because it's a very demanding and very complex plant. Because when you're comparing cannabis production with indoors, and let's say tomato production indoors, when you're producing tomatoes, you just care about the biomass and the fruit quality,
Hmm.
So we will try. You're trying to actually provide the optimal growing, conditions to the plant, uh, so the plant will have more biomass and improve fruit quality. But when you talk about cannabis, for cannabinoid production, you're producing secondary metabolites. So it's a fine line between producing biomass and producing uh, plant secondary metabolites, terpenes and cannabinoids. Um, so, and it's a well-known concept that in order to stimulate uh, plant secondary metabolites, you stress is a way to stimulate or create this trigger to increase the production of plant secondary metabolites. Because usually, like a good example that I like to give is like plant secondary metabolites. I mean, I'm saying, I'm trying to be more specific, because I saw that there are people from other backgrounds, watching the podcast, like more like medical background. So
Sure.
in that, creating a comparison with the medical side is like a adrenaline. Like, if I'm not under stress, I'm not going to be producing any adrenaline. If I'm not being stimulated to produce adrenaline, I'm not going to produce that. And it's the same thing for plants. Secondary metabolites is, as the term is saying, is secondary, it's not primary. So the plant can survive without the secondary metabolites. So it's like adrenaline. The plant need a stimulation to produce that plant secondary metabolite. And with that, there are many ways that you can stimulate the plant. Water stress, or even we are kind of debating about saying the term stress or not, because it can be more, instead of stress, it can be considered a stimulus instead of stress, because you're just stimulating the plant, you're not actually reaching that stress. And mechanical stress is one of the ways to to improve or increase this production of plant secondary metabolites. So that's why we decide to use vibration. And I decided to present this specific study at CanMed, because this other, abiotic and biotic stress, like, for example, water stress, nutrient deficiency, temperature stress, light intensity, they're more common. Mechanical stress is pretty cool, it's pretty new. So, and that's why I decided to present at CanMed that this specific study.
All right. So if I'm following you, so the so
I
the
talk a lot, I know.
no, it's great. No, but it's been said before that the cannabinoids and the terpenes, they are in some ways a defense mechanism.
Hmm.
They're antimicrobial and they may ward off pests or something like that. So with that in mind, so introducing this stimulus or this stress is actually pushing the plants to express those terpenes, those cannabinoids more in response to that stimulus that you're providing it, right?
Exactly. Because if all the condition, the growing conditions are optimal, the plant will be like, why I will be producing plant secondary metabolites? It's all good. I
Right.
don't really need it. Of course, the genetic part is also, I mean, the plant will naturally produce plant secondary metabolite anyway, but the stimulus can be like a practice or a management to increase the production.
Yeah, I mean it's like bodybuilding, right? If you want to
Yeah,
build muscles, you need to stress your muscles so that your body responds to it.
and then that's a good example when you talk about mechanical stress, because bodybuilding is also is like is a mechanical stress in your muscles. So uh yeah, that's a good correlation, and we were pretty much kind of trying to body build the plants.
All right. So how were you introducing this mechanical stress? I know you mentioned vibration. So how were you doing that?
So it's actually a collaboration with a company from North Carolina, and it's the way that this mechanical vibration is implemented in the plant. It's a like a metallic frame, and a transducer is actually transmitting 200 hertz frequency and 15 decibels amplitude to the plant. So it's actually a very subtle vibration. It's not like you're not really shaking the plant. It's a subtle vibration, and in this specific study that I presented at CanMed, it was 15 minutes per hour during the entire cycle of the plant.
Okay. And what were the results? Were there any surprises?
Yeah, no, but another mechanical stressor that we test, it call it high stress training. Uh,
Okay.
that was the other mechanical stress that we test in this specific study, and the combination of both. So um, the results were really cool, especially because there are many studies, not many, but some studies showing that mechanical vibration or mechanical stimulation can um, minimize inflorescence elongation, and that's exactly what we found in the study. So because the buds were heavier, the plants under only vibration, not high stress training, presented heavier buds, and or we are assuming that as the inflorescence elongation is decreasing, you're creating a more like compact,
Mm.
like a heavier bud, and that's what we found. Uh, Unfortunately, we didn't see difference in terms of total biomass. We thought that we would see at least for the high stress training. And again, if you don't know, do you know what is high stress training?
I think I'm familiar with low stress training, but not so much high stress.
Yeah. So high stress training is it's pretty much you bend the main cola or the apical
Hmm.
part of the plant. You bend it very carefully because you don't want also create a wound where pathogens can enter and affect the plant. So you bend that apical meristem, and so you're pretty much trying to cut that apical dominance. So and then when you bend the plant, the plant will start growing more like a reverse umbrella
Right.
instead of like Christmas tree.
Like topping
uh
it, right?
yeah, it's like topping without cutting.
Okay.
Yes, it's pretty much like topping without cutting the plant. So that's why it's more like, yeah, that's why topping is topping and high stress training is high stress training,
Makes
or
sense.
super cropping. Some people also
Yep.
call that super cropping.
Okay. So was introducing this stimulus, did you see a difference in the cannabinoids or
Oh,
the
okay, good question. So that's actually a ongoing study. I
Hmm.
have a student that now it's analyzing the cannabinoids and terpenes. So it's not done yet. So and that's the main part, the most important part. We are very curious to see, to analyze the data. But with that said, like as we didn't see any statistical significant difference in terms of total biomass. It
Hmm.
means that control, high stress training, vibration, and high stress training plus vibration, all the treatments had the same total biomass and bucket biomass. Uh, Considering that, if you see an increase in plant secondary metabolite, it means higher productivity because you're not losing biomass. Because you could, like, okay, the plant is under
Mm,
stress, you change it, we will translocate the photosynthase to other, to kind of heal from that stress instead growing. But in this specific study, all the total biomass was the same. So if it's increasing cannabinoid, it means higher productivity.
right. So that's a win. Yeah, absolutely.
Yes, but we don't know yet.
Okay,
Yeah.
excellent. Well, interesting. Interesting to see the findings there. All right, so moving on to another technique that you guys were investigating for improving production, plant hormones. Um, I don't know if I'm going to attempt to pronounce this one, but tell me a bit about. All right, I'll try.
Do it.
Methyl jasmonate.
Yes. Yeah.
Okay.
Uh, yeah. As you mentioned, methyl jasmonate is a plant hormone, and also as you mentioned before, the plant secondary metabolites or the cannabinoids are correlate, or I mean, in general, plant secondary metabolites are correlated to defense mechanisms. And methyl jasmonate specifically, it comes from the family of the jasmonate hormones. But methyl jasmonate is a very volatile plant hormone that is correlated to plant defense.
Okay.
So it's pretty much when the plant is under stress, it's the plant release methyl jasmonate to kind of inform or signal, is as a signal to the other plants, like, hey, uh, I'm under stress, something's going on.
Okay.
Like, let's say draw stress. Let's say the plant in an outdoor condition, and then there is a specific area of the field that is drying, and then the plant is pretty much signaling, like, informing the other plants around, like, hey, I'm under stress, something's going on here. So, and then that's the compound that the plant release, this volatile compound methyl jasmonate. So, and as we're working with this, stressors or stimulus, uh we were like, well, if you stress the plant, if you actually stress the plant will release this plant hormone. How about if you, like, exogenously, that's a hard word too, exogenously or artificially apply this plant hormone that is actually mimicking the stress. So, and that's what our hypothesis, like, okay, instead kind of creating a real stress, let's just fake a stress, like
Yeah,
artificially add this plant hormone and see if the cannabinoids will increase. So that's pretty much that's why we use methyl jasmonate in some of our studies.
excellent. And so what are the results? Was this successful?
Well, I mean, the thing is, what we are seeing a lot in your studies is that most of the results are very genetic or
Yeah.
cultivar dependent, very, very cultivar dependent. So, um, and also it's, the thing is, that's the tough part of, working with cannabis. For other plants, you have, more studies addressing different doses, different concentration of methyl jasmonate that you can, you know filter out and find a better dose to be more effective. But for cannabis, we can use, these studies, as a background information, like, oh, you know this range of the concentration, let's say 50 micromoles to 200 or 100 or 200 micromoles. It's a decent range for other plants, so let's use it, let's try it. but you don't, I mean, that was like, there are not many studies in cannabis. So in this trial, the results are interesting. We actually increase trichome density.
Hmm.
I have two studies addressing methyl jasmonate, uh, but in both, the trichome density increased, um, but it was not correlated to higher cannabinoid concentration.
Interesting.
So this is another topic that is interesting as well, because I'm seeing a lot that trichome density is not directly correlated to cannabinoid concentration. And this is kind of a dangerous topic to talk with, especially with legacy growers, because trichome density has been like a plant parameter that's been being used for many years as okay, more trichomes, higher cannabinoid.
Sure.
And then when we start kind of researching it and not finding it, some legacy growers, they don't really like to hear that,
Huh.
yeah.
So what's I guess I'm asking you to speculate, but what's sort of the explanation there? Because the cannabinoids are made in the trichomes, right? So you would assume if there were more of them, there would be more cannabinoids.
So that's why we actually published a paper today. It was released today, this
Oh,
morning, uh, uh, describing or discussing this lack of correlation. And there are many reasons that we can um, guess. Because it could be, it can be like a very, like, hey, no brainer, more trichomes, more cannabinoids. And that's what we have seen, or that's what we believe for many years. But the thing is, when you're counting trichomes, first thing, that's what we include in this publication. What part of the plant you're using as a sample? There are many parts of the plants that you can use as a sample. The bract, the sugar leaves, the fan leaves, the entire bud. So what part of the plant are you using? And in after you decide the part of the plant that you're using, what area of that part of the plant you count. So
hmm.
that's one. Another reason that sometimes the correlation, we are not seeing that correlation, is that um, um, when you're counting trichomes, of course, you're just going to count the globular trichomes. You're not counting, because they're the only trichomes that are storing cannabinoids. But when you're counting them, you're not considering the size of the head.
Hmm.
So what I'm trying to say is, like, two very big trichomes, or no, let's say, two very big trichomes could store more cannabinoid than five very small trichomes. So if you count five and two, there's no correlation. There's no correlation. So the shape of the trichome is also, you have to consider. There are like already research proving it, that as the tallest the trichome is, more cannabinoid it's
Hmm.
stored or concentrated. So 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 then we have to consider the size of the head. You have to consider the shape. You also have to consider that sometimes the plant is going under stress, through some stress, and the tissue, the vegetal tissue, it's expanding and shrinking, and that can also change the how the density, because you're counting by area. So, and
Hmm.
that also can change the density because of that expanding and shrinking. And then that's why there are many ways that trichome density cannot be correlated to cannabinoid concentration.
Interesting.
So, and sometimes you don't think about it,
Yeah.
you know.
Well, it makes me wonder too. I mean, which would be the better metric? Would a slightly lower cannabinoid concentration, but a more trichomes in some ways be more desirable than the opposite?
So then the conclusion of this study, we found that the bracts are the best floral structure to be used, because it's very homogeneous.
Hmm.
But more than that, it's important to use more than one floral structure to correlate it to the the cannabinoid concentration, because when you're taking the sample, you're taking the sample for the cannabinoid concentration, depending of if you're using, let's say, HPLC, is
Right.
like 1 gram of the main cola.
Hmm.
So, and it's pretty much it's not only bract or only sugar leaf. It's a little bit of everything, and that's why using more than one plural extractor will
Mm.
be more uh, reliable correlation.
Interesting.
And also another thing that is important in that we describe in that paper too is that, so okay, trichome density maybe is not the best way to correlate it with trichome concentration, but trichome density, it's a very good aesthetics
Right.
plant parameter, because this is correlated to the frosty bud.
Right.
That's
Bag
that's
appeal.
okay. We cannot negate. It's higher trichomes density is going to be more frosty buds. You
Right.
know, so it's more aesthetic parameter
Right. Of
or
course. Of
quality
course.
parameter.
Yeah, that's why I was asking, is there still something to that? The fact that there's lots of them, I don't know, but
So yeah, that's the thing. Maybe the frostier buds are not the ones with the highest cannabinoid concentration as you used to think.
mhm.
Because it's just frostier doesn't mean that the concentration is higher.
Right, right, right. All right, very cool. All right, so we covered that one kind of in a roundabout way, but
Yeah,
that's cool. But that's cool. I like how we're connecting everything. So one of the other stressors that I know that you guys had looked at was water stress and how
Mhm.
that might affect cannabinoid production. So tell me a bit about that.
Yeah. Water stress is actually is something that is well tested for many other plants in general, as a just to learn more about the physiological responses of the plant under stress.
Mm,
Uh, but when you talk about water stress or water deficit stress for cannabis, it's a very um, I would say common practice, especially when you talk about crop steering. There is a term that we call the drybacks, which is pretty much when the soil media it's drying. Is it that timing when the so you're not irrigating the plant,
hmm.
so the media is drying, and that period of that the media is drying can stimulate plant growth, cannabinoid production, uh, nutrient uh, absorption. So that's why when you talk about water deficit stress, we have to be careful about, is it a stress or is it a stimulus?
Hmm.
Because sometimes it's not enough to be a stress. It's not enough to be so harmful to the plant that to the point that it would decrease biomass or harm the plant. Sometimes it's just a stimulus to, like, oh, the water here is decreasing here, so we need to be more efficient in terms of uptaking nutrients,
Yeah.
or start producing secondary metabolites to survive or to be prepared for this stress period. So um but they study that you're talking about, the study that we published, the study you're talking about, it's a review, but we are actually working on another two studies addressing water deficit stress, and it's then we actually call water deficit stress, not water deficit stimulus or water decrease stimulus, something like that, because we really stress the plant, and we decide to do it because, again, I mean, our lab here is pretty new, so we want publication. So we want to conduct these studies that are going to present statistical significant differences.
Hmm.
And these two water deficit stress studies, one of my master's degree students, she just graduated, she conducted. And the first time she conducted a study, was a mild or moderate water stress, was only 4 to 5 days stress. And she conducted a study for 5 months, collect the data, analyze, the cannabinoid, biomass, everything, and then zero statistically significant
Hmm.
results, like zero. And it was like, I mean, you cannot publish. I mean, there's nothing to publish here. And then I told her, okay, now let's go extreme. Let's do extreme water depth stress. So, and she was so scared or concerned or worried about killing the plant. But then we realized that it's cannabis such a plastic and
Hmm.
plant, because some of the stress, it was like 12 days, 10 days, 11 days with no water, like zero water. And the plants were surviving like almost 2 weeks without any irrigation. And as soon as they irrigate, the plants were boom coming back.
Hmm.
So, and so again, that's why it was we made it, for this specific study we said water deficit stress, because it was actually a stress. Um, so we are working on a publication. I actually going to present one of these studies at the ASH, at the horticulture, the American Society for Horticultural Science, uh, next week in New Orleans. Um, okay, so two different studies. The first study, we are trying to find the best frequencies of stress, because usually the water stress is doing flowering,
Hmm.
because doesn't make much sense to stress the plant in veg, because there's no trichomes. So why, you're just gonna lose biomass.
Hmm.
But when we start flowering, that's the time to create that stimulus for more production of cannabinoids. And then we test frequencies. No stress, 3 stressors during the flowering cycle, 2 stressors during the flowering cycle, and 1 stress during the flowering cycle. And we found that 1 stress was the best compared, but kind of correlated to control. Like
Hmm.
1 stress and no stress was kind of very similar. Then we use, okay, as we're seeing that, but again, we are talking about extreme stress, extreme water depth stress. Then, as we found the one extreme water depth stress was the best, we decided to find the best timing in the growing in the flowering cycle.
Hmm.
Okay, is the best early flowering, mid flower, or late flower. So that's where the second study that she conducted. And but overall, take home message of these both two studies: extreme water stress maybe is not the best way to do it.
Hmm.
I mean, of course, we found a lot of statistically significant results, but let's say 12 days without water, it's too much.
Yeah. So you found statistical significance difference, but it sounds like it was a detriment. It was a detrimental effect rather than beneficial.
Yeah, but sometimes, at least you're learning that, it's
Sure.
not, I mean, of course, it was against our hypothesis, because we thought that stress would increase, uh, secondary metabolite, but I think it was too much, was too extreme, because we also end up losing biomass. So then you lose biomass, and the cannabinoid was not
Sure.
that high to compensate the biomass that you lose. And another thing is water stress is somehow correlated to nutrient deficiency, because as you're not adding water, like fertigation,
Hmm.
you're also not adding nutrients.
Right.
So it's correlated.
Interesting, interesting. But I mean, one of the things as a former grower who has given it up because I do not have a good green thumb, I think I was guilty of overwatering, maybe watering too often. But what I thought was interesting from the first one was, having a week's worth of missing watering didn't really have that much of a detrimental effect on the end point, right?
No, exactly. I mean, you used to be a home grower
Yeah, a
or
little
actually,
bit. Yeah,
but
no,
home,
not commercial.
no
Definitely
commercial.
not.
Okay, no, no, that's exactly the point. At least we found that, okay, I'm a home grower. I'm gonna be traveling for a week. Doesn't matter. I can leave my plants here, and when I come back, I just rewater them, and they will be okay.
Yeah.
You know, at least, and could be even almost 2 weeks that
Wow.
you can come back, and I mean, these studies are not published yet, but I mean, I will be happy to share with you because we have a lot of comparison photos,
Mm.
and you see like under stress, and then as soon as you water the plant is boom, kind of revive. So that's so cool to see how cannabis is plastic.
Yeah. It's sort of like a cat. You can go away for a week
Yeah,
and just leave
yeah,
them, they'll be fine.
exactly. Exactly. As long as you keep the lights on, you know, you don't need to worry much about water. Of course, it might affect, production, but you cannot be like, oh, no, I need someone to take care of my cats or my plants. You know, 5 days will be okay.
Awesome. All right. That's really cool. All right. Well, one last thing I wanted to talk to you about before I let you go was the standardized cloning procedure that you guys had published on, which I think again kind of speaks to that point of if you're doing research and in comparing research, you need to have a standardized protocol, right?
Exactly. I mean, in this protocol, we decided to publish. I mean, we create that protocol was like an in-home. I mean, just for our lab, because as we're doing, all our studies, we have our mother plants in our lab. We have our mother plants room,
Hmm.
because all the studies we're conducting, we are taking the clones from one single mother plant. Because
Right.
actually, I don't know if you saw one of the studies that I sent to you. We couldn't get all the clones from just one single mother plant,
Mm.
and we ended up taking from two different mother plants from the same cultivar.
Okay.
And that the mother plant effect was bigger than anything else.
Huh.
So we thought that mother plant would not be an effect. We didn't even consider as effect in the beginning. But when we ran the statistics, mother plant, the two different plants, it was showing like a lot of differences. So then it comes to a problem of
So
cloning, labeling. Go
so
ahead.
were those mother plants both clones from an original plant or were they grown from seed separately?
Clones from the same plant.
Wow, that's interesting.
Yes, that's and we found differences. And uh, so that's why we decide, okay, right now, as we are conducting the next studies is going to be all, because we don't want to create another effect. So all the clones will be from just one mother plant. And as we create this cloning protocol to be standardized throughout all the studies, and then as new students are coming, they already know what to do. It was like a extension publication. Then we decide to extend that extension publication, and uh, um, publish. You know, because we have cloning or propagation protocols for other plants,
Hmm.
but not much for cannabis. We kind of like a hearsay. People
Right.
talk about good ways and good practice, and then we decide to publish that. Because again, that's another cool thing about working with cannabis. Because many things that we're doing is not crazy or very fancy or a rocket science, but it's something that was the first time done in cannabis.
Right.
So it's the same thing for the trigolinine study. Trigolinine it's an alkaloid. It's a plant secondary metabolite that is known as a water deficit stress marker for other plants,
Hmm.
but there is no studies addressing that in cannabis. And then I'm like, oh, well, let's be the first one to do it. And then we found that trigolinine is also a good way, it's a good marker for water deficit stress in cannabis.
Yeah.
So that's another another question that I guess was that I'm
Yeah,
kind of
no,
mixing
and
around.
and it's, it's cool that you point that out, that, a lot of the things that you guys are doing, it's the first time it's ever been done in cannabis. And I think to your point too, perhaps it's taken for granted when you study other crops that there are standardized procedures for cloning, for measuring whether it's trichome density or anything like that. And the fact that you don't have that in cannabis yet makes it really hard to sort of compare results from other studies.
100%, 100%. Because some research that we're conducting in our lab, it's like it's being done, let's say 100 years ago for corn,
Right.
you know, but cannabis is a totally different plant. So maybe the responses are not the same. Of course, you can use that trend, you can use that information as a background, but it's not the same. It's a different plant,
Right.
you know. So that's why that's another challenge that my students they have when they're writing like the introduction part of their like master thesis or PhD dissertation. They don't have much studies. They don't
Yeah.
have much literature to include in their introduction because there are not many papers. So the way they solve it is adding other plants, correlating
Yeah.
that approach with other plants.
Yeah. So we need more studies. We need
Yeah. So, but
we
we
need
need more funds.
we need more funding. Exactly. All right. Well, maybe that's a good segue, Jose. So, where can people learn more about your lab, the work that you're doing, and maybe there's an angel investor out there that wants to give you some funding. How do they find out more about
Awesome.
you?
Awesome. Well, I do have a, a pretty good social media presence. I have my Instagram that is doc Leme Leme. On Instagram, I'm Jose Leme, PhD on LinkedIn. I have my website as well, drleme.com. So I try to be, post what our lab is doing, and not, I mean, as I mentioned that, is a metric for my tenure. Uh, I'm not thinking much, I mean, it's just kind of showing what my students are doing, the research we're conducting, what are you doing in the classroom with the undergrads, the new classes that I'm teaching, the events that I'm attending. Oh, by the way, we are organizing an event here at SIU
Oh, great.
on September 19th. Is the SIU Cannabis Symposium. Um, it will be really cool. So Dr. Bruce Bugby is
Oh,
a
great.
keynote speaker. Also, Dr. Alison Justice,
Oh,
she's
cool.
also a
Yeah, yeah, yeah.
keynote speaker. So, and it's a 2 days event. The first day is the symposium, more nerdy, more informational, and then the second day is the hemp hops and shrooms, which is more like a festival in Carbondale downtown with bands and vendors, and so it will be a really fun weekend.
Excellent. Yeah, no, I'm happy to put all the links to your social media. I'll link all these studies that we've been talking about, and throw a link in there too, so people can check that out. That sounds like an awesome event.
Yeah, and I will send to you the study that we just published today. So
Yeah,
pretty
please do.
interesting too. Pretty interesting too.
Awesome. Hey, this has been great, Jose. Thanks so much for the time, and yeah, keep up the good work. We hope to see you at CanMed 26.
Yes, it's gonna be in Boston, right? Or it's not?
Well, still to
It's
be
still.
determined. We're still working that out.
I remember like everybody was like very curious about okay where is gonna be the next next one. So then I heard about Boston, and then so okay, it's still up in the air.
Still to be decided. No details
Okay,
yet.
okay. No, yeah, I'll be happy to attend. I really enjoy the event. So a lot of good connections. So and again, I really enjoy this podcast. So thank you for inviting me.
No, thanks so much for doing it. It's been great.
Thank you. Thank you.